Hi, welcome to the first physiology lecture on the heart. I am Dr. Richard Francis. Let's get going. So these are the learning objectives for both of my lectures. I won't go through them now, you can look at them for when you're studying for the exam. Firstly, let's just talk about the basics of the cardiovascular system. The cardiovascular system has three basic components, which I'm sure you're all aware of by now. You have a heart, which serves as the pump that establishes the pressure gradient needed for blood to flow through the vascular system to the tissues. You have the blood vessels themselves, which are the passageways through which blood is distributed from the heart to all parts of the body and back to the heart again. And finally, you have the blood, which is obviously the transport medium within the blood vessels, which transports the nutrients to the tissues and then transports away the waste products from the tissues. The circulatory system is basically divided up into two parts. You have the pulmonary circulation and the systemic circulation. The pulmonary circulation is a closed loop of vessels carrying blood between the heart and the lungs. So obviously deoxygenated blood goes from the heart to the lungs to get oxygen, which makes the blood oxygenated, which then goes back to the heart. You then have the systemic circulation, which is a closed circuit of vessels for carrying blood between the heart and other body systems. So the systemic circulation takes the oxygenated blood that has come from the lungs, delivered to the heart, and then the heart will pump this blood to the body where the oxygen is taken up by the tissues, waste products are then absorbed, and then this goes back to the heart. So the heart itself is a fairly complex structure. You can see here this is a basic cartoon of its anatomy. You have two sides, left and right side. Usually when we look at the heart, we say the left side is on the right side, because we're basically looking at an individual face on. This individual is looking at us, and their left side is on our right. This is usually how it's presented. So the left side of the heart, carrying oxygenated blood from the lungs to the body. This oxygenated blood arrives in the pulmonary veins, enters the left atria, and then the blood goes from the left atria into the left ventricle. The ventricles are the muscular compartments responsible for generating the force of ejecting the blood through the system. So these are the thick-walled muscular chambers. Left ventricle then ejects blood out through the aorta. The right side of the heart is the same, but slightly different. You have blood returning from the body, deoxygenated blood, going into the right atria. This then delivers blood into the right ventricle, which then pumps it through into the pulmonary arteries, which delivers this deoxygenated blood into the lungs for getting more oxygen. So you can see here, just off this figure, there are two important sets of valves. You have the large Atrioventricular valves, which lie between the atria and the ventricles. The specific names are given to them. On the left side of the heart, these are called the bicuspid, and on the right side, it's called the tricuspid. Or you could just refer them to the AV valve, atrioventricular valve. And then you have two sets of valves between the ventricles and the arteries. These are the semilunar valves, obviously, which stop blood flowing from the arteries back into the ventricles. Now, this diagram is obviously fairly complicated. It's maybe much easier for you to remember the main structures of the heart if you look at the heart like this. The heart is basically two pumps side by side, the left side and the right side. On the left side, you have blood coming from the lungs to the left atrium through the mitral valve, which is the AV valve, atrioventricular valve, going into the left ventricle, and then blood from the left ventricle is pumped out into the aorta through the aortic valve or the semilunar valve. Similarly, on the right side, you have blood coming from the body through the vena cava to the right atrium. It goes through the tricuspid valve, which is the AV valve on the right side, into the right ventricle, which then generates the force to eject blood into the pulmonary circulation through the pulmonary valve, which is a semilunar valve between the pulmonary artery and the right ventricle. Now the heart itself is made up of three layers, the heart wall. You have endocardium, which is a simple squamous epithelium lining the inside of the heart. You can see here in the diagram here. It's very thin. It is continuous with the endothelium of the great vessels as well. So it's a very thin layer. The myocardium is the thickest layer, especially in the ventricles, and this is the muscular wall of the heart containing the cardiac muscle tissue. It also contains blood vessels and nerves. Blood vessels obviously to supply this muscular tissue and nerves to help innervate this muscular tissue. And then the outer layer is called the epicardium, which is the outer surface of the heart. It's a serous membrane of mesothelium and underlying areolar connective tissue. Now, the pericardial sac surrounds the heart and stabilizes the position of the heart and associated vessels. And the pericardial sac is basically a way of imagining this. It's if you take a balloon and put your fist into the balloon, you basically end up with something that approximates a pericardial sac. You can see it has an inner wall and an outer wall, and between these inner and outer walls is a space. So this is the pericardial sac. So this is obviously made up of the inner wall, which is the epicardium. So this is the inner wall of the sac. It's called the epicardium, and we've already talked about that in the previous slide. And then the outer wall is the parietal pericardium, which is the outer wall made up of collagen fibers, mesothelium, and areolar connective tissue. And in the middle, in between these two layers, is the pericardial cavity. This is the space between the parietal pericardium and the epicardium. And this cavity is often filled with fluid, which helps to act as a lubricant, and it reduces the friction that occurs during each heartbeat, and it prevents these two layers rubbing against each other, generating friction, which can cause injury. Obviously, some disease states occur when there is friction between these two layers. Now, the cardiac muscle is arranged in concentric layers within the myocardium. So the atrial myocardium muscle bundles wrap around the atria in a figure 8 arrangement encircling the great vessels. And you can see here in this diagram the muscle bundles wrapping around the atria and the great vessels. The ventricular myocardium muscle layers spiral around and between the ventricles towards the apex. So you can see how these muscle layers are spiraling around. And this is because this arrangement allows the ventricles to eject blood better than if it was just parallel fibers. So when the ventricles contract, they're contracting and the muscle walls are actually working like you're wringing out the blood from the ventricles. So because the muscle layers are spiraling around, when they contract, it's like you're taking a rag and you're wringing the liquid out of a rag. And this is exactly what's occurring here. The ventricular myocardium is contracting in a way to get the most ejection of blood out of the ventricles and into the arteries. Obviously, the heart muscle works very hard and it's essential for pumping the blood, so the heart muscle needs its own blood supply. And this blood supply is called the coronary circulation. And it's because the myocardium is too thick to allow blood to diffuse from the ventricles themselves into the muscle layers. So you need to have your own blood supply to these muscle layers. And the vessels you should make sure you're aware of are the left and right coronary arteries. These are the two main arteries branching off from the very base of the aorta, and these then supply all the other blood vessels supplying the heart. And then the veins all drain in to a main vein called the coronary sinus, which then drains into the right atria. So those are the main vessels you should be aware of. I'm not going to be expecting you to remember all these other things here, but you should know there are two arteries that supply the heart, left and right coronary arteries, and the blood then drains through the venous system of the heart to the coronary sinus. Now, there are structural differences between the left and right ventricles. You may already be aware of this, but just looking at this cross-section, you can see straight away the left ventricle is very thick, the right ventricle is a lot thinner. You can see here differences between dilated and contracted stages of the ventricles. And you may already know why the left ventricle is thicker than the right ventricle. So the ventricular muscle, just like any muscle, if it works, if you exercise it, it'll get bigger. So the left ventricle is bigger because it works harder than the right ventricle. Just like if you're lifting weights, your arm muscles will get bigger if you're doing more work with them. The left ventricle does more work than the right ventricle, so the left ventricle is thicker. And the reason why the left ventricle is thicker is because it has to eject blood at a higher pressure. So you can see here, this is the whole circulatory system laid out in a straight line, showing you different locations across it. So a blood cell going through a whole circuit would go through the left atrium, left ventricle, aorta, down through the systemic circulation until it returns back to the right atrium, goes to the lungs, and then back through the lung vessels and the pulmonary veins, and then back to the left atrium. So this is one single circuit for a blood cell through the system. And you can see here straight away the aorta is very high pressure. And this means the left ventricle has to work a lot harder to eject blood into this high pressure systemic circulation. And they call this work afterload. We'll be talking about what afterload is in a future slide. For now though, you should be aware that obviously the systemic circulation is much high pressure, where the pulmonary circulation is low pressure. You can see here on the y-axis the actual pressures are given here. So whereas the systemic circulation, you have systemic pressure around 120 over diastolic pressure around 80, pulmonary circulation much less than that. So this means the heart, or specifically the left ventricle, has to work a lot harder to eject blood into this high pressure system. And this is obviously why the left ventricle is thicker than the right ventricle. It has to do more work to eject blood into the aorta than the right ventricle does when it has to eject blood into the pulmonary artery. Now the cardiac cycle. So the cardiac cycle describes the events that occur from one heartbeat to the next. And this is showing you a heart in someone's chest. I think it's just before a heart transplant. You can see the ventricles and the atria are contracting and relaxing. So a general term is diastole. Diastole is used to refer to the period when the heart muscle is relaxed. So obviously there's slight differences between ventricular diastole and atrial diastole because the atria and the ventricles contract and relax at slightly different times. Generally speaking though, diastole is referred to when the heart is relaxed. Systole is a term used to describe the period when the heart muscle is contracted. So once again, you have slightly different times when the ventricles are contracted versus when the atria is contracted. So ventricular systole versus atrial systole is slightly different times. So coordinated contraction of the atria and ventricles is essential for normal heart function. So it may be a little hard to see in this movie here, but the atria are contracting first and then the ventricles are contracting. And this is shown in the diagram down below, the timeline showing the left side and the right side. And you can see straight away, atrial contraction occurs first and then ventricular contraction. So this coordinated contraction is essential to allow the heart to function normally. Now let's go through a complete cardiac cycle and we'll see exactly what's occurring during a full circuit. So we'll start at the very beginning. This is when the heart is completely relaxed. Both the atria and the ventricles are in diastole, which means both atria and the ventricles are relaxed. So both chambers are relaxed and the ventricles are filling. So blood is rushing through the veins into both atria, and then blood is going from the atria passively into the ventricles. So this is all a passive process. Blood is rushing through into the ventricles. After that, you get atrial systole, and this is when the atria contract first. So you can see here this is represented on this diagram, atria labeled in red. The atria contract to push more blood into the ventricles. This allows the ventricles to be topped up with blood. And this accounts for only about 10 to 20% of the total blood in the ventricles. So most of the blood that is entering the ventricles occurs during this passive filling stage during diastole. And after this, the atrial contracts to top up the blood volume in the ventricles. Following this, the ventricles will start to contract, and they call this phase isovolumetric contraction. And during this phase, because the ventricles are starting to contract during ventricular systole, this pushes the AV valves closed. So the AV valves are closed, but the pressure in the ventricles is still not enough to force blood out into to the arteries. So these semilunar valves are closed as well. So this is why it's called isovolumetric. Iso being the same, volumetric being volume. So you have a volume of blood in these ventricles, and this volume doesn't change. The ventricles are starting to contract. This shuts the AV valves closed, preventing blood from leaving the ventricles back into the atria. But the pressure in the ventricles is not enough to open the semilunar valves. So the pressure builds up in the ventricles during this isovolumetric contraction phase. Then, once the pressure in the ventricles exceeds the pressure in the arteries, this allows the semilunar valves to be pushed open, and you get ventricular ejection of blood from the ventricles into the arteries. And this obviously also during ventricular systole, the ventricles are still contracting. So isovolumetric ventricular contraction occurs at the beginning of ventricular systole, then when pressure is built up enough to force open the semilunar valves during the rest of ventricular systole. Then after ventricular systole, you get the heart ventricles relaxing, and you enter a phase called isovolumetric ventricular relaxation. Once again, remember isovolumetric, iso meaning the same volume, volumetric meaning volume. The volume in the ventricles remains the same because both sets of valves are closed, and this is when the ventricles are relaxing and the pressure is dropping in the ventricles. This means these semilunar valves are snapped shut to prevent backflow of blood from the arteries back into the ventricles. And because the pressure in these ventricles is still higher than in the atria, the AV valves haven't opened yet. So pressure is falling in the ventricles during this phase until the pressure falls below the pressure in the atria, in which case the AV valves will then open, allowing blood to flow from the atria into the ventricles. So this is one single cardiac cycle. So before we go on, we need to define a few terms that are commonly used when talking about cardiac physiology. Firstly, end diastolic volume, or EDV. Now this is the volume of blood in the ventricle at the end of diastole. So remember diastole being when the heart muscle is relaxed. So just before the heart starts to beat or contract, you have a volume of blood in the ventricle, and this is called the end diastolic volume. And also just be aware, if I highlight something in red here, you need to make sure you understand the meaning of the term or what's being talked about. Okay, so it's a very high probability this may come up in the exam. Similarly, we have a volume called end systolic volume. In this case, this is the blood volume in the ventricle at the end of systole, so at the end of the heart beat, the heart contraction. So you may be surprised to see that there's still blood left in the heart, in the ventricle after the heartbeat. And under resting conditions, it can be quite a lot, 60 mils, you can see here. And if we're exercising and we stimulate the heart to beat stronger, this volume is going to fall a lot less as the heart pumps a lot more blood out after each heartbeat. But make sure you understand, at the end of each heartbeat, there's still going to be a small volume of blood left, or even a large volume of blood. And this is called the end systolic volume. And this is how we calculate stroke volume. And stroke volume is the total volume of blood pumped out from the left ventricle per heartbeat. So to calculate that, obviously you take end diastolic volume, the total amount of blood in the ventricle at the start of the heartbeat, subtract from that end systolic volume, which is the volume of blood at the end of the heartbeat. And you're left with the total amount of blood pumped for the heartbeat, pumped during the heartbeat. Next we have heart rate, so this should be fairly straightforward for all of you. This is the number of times per minute that the ventricles eject blood. I mean, you could talk about heart rate using whatever unit you want, but generally speaking we use beats per minute. It's obviously the same as cardiac cycles per minute. And finally, cardiac output. This is a term used to describe the amount of blood the heart pumps per minute. So obviously to calculate cardiac output, you'd need to know the heart rate, and you'd need to know the volume per heartbeat, which is stroke volume. So make sure you understand this equation. Cardiac output equals stroke volume times heart rate. And using the values we've put in above, this equals about 5 litres per minute. Okay, and blood pressure is the force per unit area of blood pushing against the vessel's walls that contain it. Two things determine blood pressure: cardiac output and peripheral vascular resistance. And generally we use this equation here. Blood pressure is determined or equaled cardiac output times total peripheral resistance. So obviously cardiac output is the amount of blood being pumped out of the heart, and peripheral vascular resistance is the resistance to the blood running through the blood vessels. And also remember, cardiac output equals heart rate times stroke volume. Now, the heart sounds. There are two heart sounds you would normally hear with a healthy individual. They're called S1 and S2 heart sounds. If you haven't already heard a heartbeat, maybe go on YouTube and listen to it. Now, the first heart sound is caused by the AV valves snapping shut. So remember, blood during cardiac diastole, blood moves passively from the atria into the ventricles. Then when the ventricles start to contract, it pushes these AV valves shut, and this causes the first heart sound. So the S1 heart sound, or the first heart sound, is caused by these AV valves snapping shut, which occurs at the start of the heartbeat when the ventricles start to contract. The second heart sound, or the S2 heart sound, is when the semilunar valves are closing. So this occurs at the end of systole. So during systole, the ventricles are contracting, generating pressure in the ventricles, which forces open the semilunar valves, injecting blood into the arteries. And then when the pressure in the ventricles falls below that in the arteries, this causes blood to flow backwards, snapping shut the semilunar valves. This causes the second heart sound, or the S2 heart sound. Now, we've talked about the cardiac cycle and the physical events that occur during a single cycle, and these physical events are caused by electrical signaling within the heart. So the heart beats rhythmically as a result of action potentials it generates itself. It's autorhythmic. These action potentials precede the mechanical events. So first you have an electrical signal going through the cardiac cells or the cardiac muscle, and then you have a mechanical event, which is the cardiac muscle contracting during systole. Now there's two specialized types of cardiac muscle cells. You have the contractile cells, which is 99% of the cardiac muscle cells, and these cells are the ones that do the mechanical work of pumping. These are the muscle cells that will contract. These muscle cells do not normally initiate their own action potentials. So 99%, these are by far the most of the muscle cells, are contractile cells. Very similar to the skeletal muscle cells. Then you have about 1% of the cardiac muscle cells being autorhythmic cells. These autorhythmic cells don't contract. Instead, these are specialized for initiating and conducting action potentials responsible for the contraction of the working cells. So these are forming, I guess you could say, the wiring, electrical wiring of the muscle, the heart muscle, sending the signal through the heart muscle to help coordinate the contraction. So the cardiac conduction system is formed by the autorhythmic cardiomyocytes that specialize in action potential generation and conduction rather than contraction. So there's a number of different structures you should be aware of. This Sinoatrial or SA node, which is also known as the pacemaker. I think most of us have heard of the pacemaker. This is where the signal originates, then it travels through the heart, through the atria, then into the AV node, or the Atrioventricular node, and it goes through the Atrioventricular bundle, also known as the bundle of His, down through the interventricular septum, and then up through the sides of the ventricular walls through the Purkinje fiber. So the sinoatrial or SA node generates the impulses that sends a signal throughout the rest of the heart. And under resting conditions, it generates impulses at about 70 to 80 times per minute. And it can generate these without any external stimuli. If you remember back to the movie we saw, the few slides ago, we saw the heart beating isolated without any other input to it. So this is all due to the SA node generating impulses around 70 or 80 times per minute, which then spread throughout the heart. The atrioventricular node delays the impulse sent from the SA node a little bit by approximately 0.1 seconds. So the signal arises from the SA node and travels through the AV node, which slows it down a little bit. And then the impulse passes from the atria to the ventricles via the atrioventricular bundle, also known as the bundle of His, which splits into two bundle branches in the interventricular septum, and it travels up the walls through the Purkinje fibers, which carry the impulse to the heart apex and the ventricular wall. So that's the overview of the wiring circuit of the heart. You can see how it's highlighted in red. Make sure you understand and remember the main points along this pathway. Now let's look at this pathway a little bit more in depth. So the sinoatrial SA node is located in the wall of the right atria. It's autorhythmic, around 70 to 80 beats per minute. And this obviously can be modulated from external stimuli such as nerves or hormones. But if you were to remove the heart completely from the body, it would still generate an autorhythmic pulse around 70 to 80 beats per minute. This impulse, this electrical signal, leaves the SA node and travels through internodal pathways within the atria. So it will stimulate the atria to contract during atrial systole. And you also have a signal going through the AV node. Now there's a dense layer of non-conductive fibrous tissue between the atria and the ventricles. And this prevents any signal from going through the atrial wall into the ventricular wall. This electrical signal has to go through the AV node. The AV node is acting like a gateway for the signal to go from the atria into the ventricles. It's obviously important for allowing coordinated contraction. You want the atria to contract first, and then you want the ventricles to contract second. You don't want the signal just going through the walls of the atria into the ventricles, because it would then lead to abnormal signaling, abnormal contraction. So the AV node is a node of autorhythmic cardiomyocytes embedded within the floor of the right atrium. It is, as I've already mentioned, it's a gateway for the electrical signal to go through from the SA node to enter the ventricles. It delays the action potential by about 0.1 seconds, which means it takes about 0.1 seconds for the signal to pass through the AV node. So this may seem like a small amount of time, but it's enough time to allow the atrial to contract first, and then the ventricles contract second after the atria. Remember, the atrial contraction allows blood to be pumped from the atria into the ventricles. If you didn't have the atrial contracting first and the ventricles contracting second, then obviously the blood wouldn't be moving in the right direction. AV node does have its own autorhythmicity as well, without any external stimuli. It's about 40 to 60 beats per minute. So obviously, because the SA node beats faster than that, the SA node controls or sets the rate for this signaling. But if there was some problem with the signal moving from the SA node to the AV node would then take over as a pacemaker as well. So, as I've already mentioned, delay is important so the atria can contract fully first before the ventricles. The other components of this conduction pathway are the bundles of His, which we already mentioned. This carries the impulse towards the heart apex. There's two bundle branches located within the interventricular septum. The left bundle branch is usually much larger because the left ventricle is larger. And this as well has its own autorhythmicity. In this case, about 20 to 40 beats per minute. So if there wasn't any signal coming from the SA node or the AV node, the bundle branch would then start serving as its own pacemaker. Then finally, you have the Purkinje fibers, which carries impulses into the heart apex and ventricular walls. They're located within the ventricular myocardium. And also, you can see these also have its own autorhythmicity. They can generate their own action potentials at about 15 beats per minute. And you can see this is much lower than any other point along the conduction pathway. So if there was any problem with the signal reaching the Purkinje fibers from the SA node, AV node, or bundles of His, the Purkinje fibers would then start to serve as their own pacemakers. So you can see here, this is summarized in this table. You can see different autorhythmicities from the different locations down this conduction pathway. The fastest signal will set the pace for the rest of the heart. So this conduction pathway only moves from this direction, from the SA node down through into the Purkinje fibers, because the SA node is the fastest. If there's any blockages anywhere between these different locations, then the subsequent locations can then start to take over their own signal, their own heart rate. So the SA node fires more rapidly, thus it normally controls the heart rate, thus it normally sets the pace for the heart rate. So this is the route that the action potentials take, or the electrical signals take, during each heartbeat. Now let's look at the action potentials themselves. So you've already been taught about action potential in skeletal muscle and nervous tissue. And the action potential in those tissues is very straightforward, very quick, it's very biphasic. You have resting membrane potential, then you have depolarization, peak, repolarization. And this moves along the cell membrane of the skeletal muscle membrane or the nerve axon. You get this action potential moving down like a wave of depolarization. It's very quick. It's caused by ions moving in and out through the membrane. And this is the cardiac muscle cell action potential. And you can see straight away there is a difference between the cardiac muscle cell and the skeletal muscle or the nervous axon action potential. And the main difference, you still have a depolarization, you still have a peak, and you still have a repolarization, but you now have this plateau phase in between, which greatly extends the time or duration of this action potential in the cardiac muscle cell. So what causes this? Well, first of all, hopefully we're all fairly aware that in nerve or skeletal muscle cell action potentials, the depolarization phase is due to opening of sodium channels and sodium influx through the cell membrane into the cell, whereas the repolarization phase is caused by opening of potassium channels and potassium moving out of the cell, repolarizing. So remember, resting membrane potential normally inside of the cell is more negative than the outside. If you open up sodium channels and sodium moves in, this positive charge moves into the cell, which makes the inside of the cell more positive. And then if you want to make the inside of the cell more negative again, the cell opens potassium channels, which means positive charge moves out of the cell, which makes the inside more negative again. So the cell potential, the membrane potential goes more negative again. So this, as I've already mentioned, is what you would see in nerve or skeletal muscle action potentials. Cardiac cell action potential, remember, is different as it has a plateau phase. It is similar to the nerve or skeletal muscle action potentials in that the depolarization is caused by sodium influx, the repolarization is caused by potassium efflux, but this added plateau phase is caused by a movement of a third ion, and in this case calcium influx. So calcium channels open, calcium, which is a positive ion, moves into the cell, which means the inside of the cell remains positive. So the action potentials of cardiac contractile cells exhibit prolonged positive phase called a plateau phase. This is accompanied by a prolonged period of contraction. So it's important that you have this prolonged plateau phase to allow a prolonged period of contraction. And this prolonged contraction phase ensures adequate ejection time, or adequate time to eject blood from the ventricles into the arteries. Make sure you understand that this plateau phase is primarily due to opening of calcium channels and calcium moving into the cardiac muscle cell. So why is this action potential of cardiac muscle significantly longer than that of skeletal muscle? Well, if you look at skeletal muscle, you can see in red the action potential and in blue the contraction or the muscle tension. The action potential is very biphasic and it's very quick, very short refractory period, and the contraction is much longer in comparison to the action potential. So you get a very quick signal, which is the action potential, then you get the long contraction. Now if we look at the cardiac muscle cells, you can see the cardiac muscle action potential is long due to this plateau phase, and it's about as long as the contraction or the muscle tension phase. So this means you have a very long refractory period. So what occurs in skeletal muscle? Well, if you get a lot of stimuli repeatedly stimulating the skeletal muscle, hopefully you remember you get things called summation and a tetanus. You get summation of signals, means the contraction of the skeletal muscle builds on top of each other. It doesn't have time to fully relax until when you're stimulating it so fast, you just get a continuous contraction of the skeletal muscle. Now, as you may imagine, if this was to occur in the heart during each heartbeat, it would be bad news. The heart would not be, the heart muscle, the ventricles would not be relaxing, and if it doesn't relax, you're not going to get blood going into the ventricles. So if you get a tetanus in the ventricles, basically you're going to be screwed. You're going to get no blood being pumped. So this is exactly why you have this long refractory period, this long plateau phase in the skeletal muscle action potential. This means even if you have repeated stimuli, you can see down here in these grey triangles, this is where we're trying to continue to stimulate the heart with more action potentials, whoops. But the heart is not summating these with the cardiac muscle contraction. You get a single contraction of the ventricles and then relaxation. It's only when the ventricles are basically completely relaxed when another stimuli will be successful to generate another ventricular contraction. So obviously it's important to have this long refractory period, which is caused by this long plateau phase, because we don't want summation or tetanus of the myocardium. The long refractory period means summation or tetanus of the cardiac muscle is impossible. This ensures alternate periods of contraction and relaxation, which is essential for pumping the blood. Obviously, you need the heart to fill the blood, then pump the blood, then fill the heart, fill the blood, and pump the blood. So you can't have summation or tetanus of the heart muscle. So you can see how this is highlighted in red. Make sure you understand these points. The action potential on cardiac contractile cells has a prolonged positive phase called a plateau phase due to opening of the calcium channels and calcium influx. This causes a prolonged period of contraction, and this ensures adequate ejection time from the heart chambers with no summation or tetanus. So this is a summary of skeletal muscle and contractile myocardium action potentials. So both skeletal muscle and cardiac myocardium have negative resting membrane potentials. There's slight differences, but don't worry about the differences here. Both have a depolarization phase caused by opening of sodium channels and sodium entry or influx into the cells. So if positive charges are moving into the cells, then the inside becomes more positive. This causes the spike in both skeletal muscle and the contractile myocardium. Then the repolarization phase is the same for both as well. It's caused by opening of potassium channels and potassium efflux, or potassium moving out of the cells. So once again, potassium is a positive charge. So if that moves out of the cells, you have the inside becoming more negative. But in the case of the contractile myocardium, we have a plateau phase, which is not seen in the skeletal muscle. And this plateau phase is caused by opening of calcium channels and positively charged calcium moving into the cell, keeping the inside of the cell positive. Then the duration of the action potential, it's very short for the skeletal muscle, only 1 to 2 milliseconds, whereas it's very long for the cardiac myocardium. And this long period of this action potential allows for a very long refractory period, allowing the muscle to contract without any summation or tetanus. So this refractory period is very short in the skeletal muscle, and it's quite long in the contractile myocardium, and it's due to resetting the sodium channels being delayed until the end of the action potential. You don't really need to know the specifics, but you need to know it's a long refractory period to allow the heart muscle to fully contract and then fully relax before the next stimuli can cause another contraction. And that's it for the first lecture.